So thank you everyone for joining us today. I'm Tanya Berardini. I'm from the Terra database and Phoenix Bioinformatics. I'm really happy to introduce Dr. Chris Mungall, currently head of the Biosystems Data Science Department at the Lawrence Berkeley National Lab and one of the co-PIs of the Gene Ontology project. I encourage you to read about Chris's many accomplishments in his LBL profile, as I will not be repeating more of that here. Instead, I'll kind of maybe a little history. So Terra has been involved in the Go project since 1999. And as I was digging for like, can I find the first email from Chris? I found it, May 2002, which means that we have worked together on Go for over 20 years. We have spent countless hours on conference calls, Blue Jeans, do you remember Blue Jeans? WebEx, GoMeeting, GoToMeeting, Zoom calls, as well as conferences and meetings all over the world, as he has furthered the cause of biocuration, Go annotation, and ontology development, among others, at all of these places. He has seen Go transition from editing flat files in a text editor in like the prehistoric times to using OboEdit and then Protege. Go has adopted logical definitions that further enable reasoning and move from single line gene product annotations to GoChem models, which I think we'll hear about today. And beyond Go, I think he has his finger in every single pie in the biontology world that you can think of. But here's a little known fact. Many moons ago, pre-Go, he worked at the Roslin Institute in Edinburgh on the ArcDB, which are databases for farmed and other animals. And one of the applications that he wrote in the mid to late 1990s is still up and running. You could find it if you look. I have seen him go from short hair to dyed hair to a ponytail, from clean shaven to bearded, from Mr. to Dr. Mungall, from bioinformatics scientist at Flybase to co-PI and department head. I am still amazed, but I'm no longer surprised by the breadth of his knowledge that he has accumulated in so many different areas. You know, kind of pretty much name it, he knows something or a lot about it. It's knowledge that he grows and that he very generously shares, and he will now share some of that knowledge with us. So welcome, Chris.
Thank you, Tanya, for that lovely introduction. That's great. Yeah, I can't believe it. It's been over 20 years now we've been working together. And yeah, as you say, yeah, before that, I was actually working in agricultural genetics. So it's really kind of fantastic to be kind of like speaking to this group today. I've been following AgBioData's progress over the years. I really love everything that you're doing, and I'm keen to stay involved. You know, my division right now is very interested in things like plant microbial interactions and generally kind of like healthy soil microbiome communities. So it's great to be making this connection. Okay, so I'm going to be talking about the gene ontology, and I'm going to try and give it a bit of a slant towards kind of functional annotation for plants and animals of relevance to agriculture, and how do we make all of this data fair. So you know, I think many of you are probably quite familiar with the gene ontology, but you know, I think many of us, you know, have knowledge of kind of patches of the Go, and there's maybe little kind of gaps in our knowledge. So I hope to kind of like give a general kind of big picture of the Go and go into some detail about how annotation works and how evidence works. Talk about enrichment analyses, and then some new developments in Go, such as causal activity models. And then I know because this group has a good kind of strong kind of fair data slant, you know, just a little bit about how we access the gene ontology computationally. So yeah, the big picture here, what I'm really interested in is how do we represent knowledge in a way that is accessible and machine interpretable. As biologists, we of course love to publish our results in the literature. We communicate with other biologists by writing text or making nice little cartoon figures that kind of show interactions of genes, you know, interactions of gene products with cellular components like cell walls, biological processes, and so on. And this is great for human communication, but you know, we need help if machines are able to, you know, we want to make machines able to digest this knowledge and make use of it. I know there's been a lot of progress in AI and language models and natural language understanding, but there's some nice toy examples out there. But I think these tools really have a very long way to go before they're even remotely capable of digesting some of the much more nuanced knowledge in the literature as it pertains to functional genomics and systems biology. And this is really where ontologies and the gene ontology come in. It provides a framework for semantic systems biology. So at its core, the Go consists of about 45,000 different terms or classes. And this is the bedrock that is used to support 750,000 experimentally supported annotations, totaling about 140,000 publications. On top of that, there's about 1 billion computed annotations. And all of this is aimed at making all of these wonderful results in the literature fair, findable, accessible, interoperable, and reusable. So I'd like to see Go as part of a sort of like pipeline of going from genomes to functions. So I know many people in this group are participants within the Apollo project as well, which seeks to go from raw genome sequences to being able to annotate those gene structures. From those gene structures, we can then look at the underlying gene products and annotate them with functions using the Go. So I like how these two things kind of like join together here. But just jumping right into the basics of Go. So as I'm sure many of you know, Go provides the terms for annotation of genes and gene products, and these terms are arranged in 3 different hierarchies. So on the one side, we have the molecular function hierarchy that describes molecular activity of gene products, things like DNA ligase activities. These terms are arranged in a hierarchy, going from very narrow specific terms like DNA ligase activity right the way up to catalytic activity. And we can use these to annotate the function of genes such as the CDC9 gene from yeast. Go also includes a hierarchy of cellular components. We use this to describe where the gene product is active, not necessarily exactly where it localizes to, but specifically when the gene product carries out its function, whereabouts in the cell or outside the cell is it carrying that activity. And this is also arranged in a hierarchical structure, but it's also a partonomy as well as the subclass hierarchy as well. So we have things like nuclear membranes and nucleoplasms are part of the nucleus, which are themselves cell parts. So a few less terms in this branch of the ontology, with the bulk of the terms being used to describe the coordinated activities that that series of molecular functions carry out as an ensemble. So there's about 30,000 terms here, encompassing many different aspects of cellular biology, physiology, including processes like DNA replication, biosynthesis, and so on. And each of these hierarchies goes some way to describing different aspects of gene function, what the gene is doing directly, whereabouts it's doing it, and what's the overall biological objective that the gene is carrying out in concert with other genes. And these annotations together specify a model of biological systems. So you can think of placing these annotations together to give a more detailed, coherent picture of how biological processes like DNA-directed DNA replication happen within cells. And we'll come back to this figure later on and talk about exactly the kind of like the causal flow between these different activities. But just to kind of like take you through some of the basics of Go, first of all, we can break down what exactly it means to be a Go term. Each, like all ontologies in OBO, the ontology itself follows FAIR principles. Each term has a unique persistent identifier. It has this identifier is independent of the label. The names of things can change over time. Terminology changes, but the concept remains the same. And we have a complete identifier life cycle where identifiers can be obsoleted and replaced and so on. And each term within the ontology has a textual definition that is intended to be read and understood by biologists, in particular by curators who are applying these terms to annotation. And also we provide synonyms as well for each of these terms. And these terms are arranged in a graph-like structure where they are connected to other terms via relationship types, such as is-a and occurs in. So we can take a closer look at some of these relationship types and think of the overall structure of the Go relational graph. You can download these different versions of Go from the site. There's a number of reasons why we provide these different versions, and that's because different groups want to take into account some of these other relationship types. They're maybe a little bit confusing to some users of the Go. So we produce something called Go Basic, and this includes just the most basic relationship types that typically only hold between terms within each of these three hierarchies. So we have is-a relationships, part of relationships that many of you are familiar with for relating either parts of cells to one another, so nuclear membranes being part of the nucleus, or DNA-directed DNA replication, being broken into the different subparts. But what maybe some people don't appreciate is we also link across these different sub-ontologies as well. So we have, for example, occurs in relationships that can be used to connect molecular functions and biological processes to the cell component, or even more broadly to cell types and to anatomical structures like roots and leaves in which those processes occur in. Then these processes can be causally connected via a family of regulation relationship types that I actually worked on with Tanya. And then there's a whole family of other relationship types that I'm not listing here, but I'm just providing two as exemplars, has primary input and has primary output. And these can be used to link biological processes such as biosynthesis and catabolism to the underlying chemical entities that are catabolized or metabolized. And so, of course, Go is used primarily for annotation of genes and gene products. We can break down some of the different aspects of an annotation here as well. I like to think of this as being broken down into a representation of the biology itself, and then a representation of the evidence that leads us to these conclusions. So on the one hand, we might have an annotation that says CDC9 enables the activity of DNA ligase. So you'll notice here I've included this relationship type in here, enables. This is not something that we've seen historically in some of the classic ways of viewing the Go, but it's something that we are starting to produce now in our annotations, and we're encouraging the community to make use of, because it allows more precision in how we connect gene products to the terms that describe them, in particular when it comes to biological processes. So we'll come back to that in a bit. And then the other part is crucial, which is providing detailed evidence about how we arrived at this conclusion. So this is where things like evidence codes from the evidence and conclusions ontology eco come in. So a DNA ligase annotation may be supported by direct assay evidence. And of course, experimental annotations should be supported by a publication, typically something in PubMed, such as this paper over here. So I'll walk you through some of the different types of evidence used in the Go. So the evidence behind the scenes is actually organized in a hierarchy. That hierarchy is provided by ECO, the evidence and conclusions ontology. And the top level of this ontology kind of breaks down the different broad categories of evidence that we use within the Go. So I'll walk through some of these. The primary type is the primary way of kind of like breaking this down is experimental versus non-experimental annotations. And you can see on the Go site how this the number of annotations breaks down along these lines across different species. What we typically see is something like, kind of like a 50-50 split between kind of experimental and non-experimental annotations. But this does vary as we move to other species that that maybe are not as experiment rich. So I've highlighted Arabidopsis and rice here as well. Sorry, I forgot to highlight chicken here, the fourth one from the right, Gallus gallus, which is maybe of interest to people here. But you'll see, the model system like Arabidopsis, of course, will have a lot of experimental annotations. But as you move out to other systems, maybe or less well-studied systems, you see this is less rich, and we rely more on non-experimental propagated annotations, and I'll come to those. But first, I'll walk you through some of the different experimental annotation subtypes. So these types are used for linking annotations to direct experimental evidence. So we have a very broad grouping just inferred from experiment, but we prefer to use more specific types such as IDA, IPA, INP. So for example, IDA is a common one, and this is the association between a gene product and, for example, subcellular localization, as might be determined by some kind of assay like immunofluorescence. But this is not the only experimental type as well. Obviously things like genetic interactions and mutant phenotypes, for example, gene knockout experiments, can also be used to derive Go annotations, although there are some caveats involved in those. So one of the challenges here is that Go is, it's a large consortium. We take our experimental annotations from multiple different curated databases. Each of these curated databases has their own team of curators that are dedicated to their particular organism or system of interest. And one of the challenges for the Go is how do we work with this broader consortium and come up with ways of essentially having standard annotation consistency, standardized guidelines across this, such that data is comparable and transferable. So we've worked on a number of different computational ways of doing this. So we have a system called Go rules, and this essentially provides a set of computational guidelines as well as curator guidelines on how to do annotation with the Go. And we actually have this QC dashboard that shows us cases in which, some of these rules, and these are not necessarily all hard and fast rules. Some of them are more soft guidelines, but this dashboard gives us a picture of where we need to work with individual groups to try and kind of like guide them in annotation best practice. This slide here, I think, is maybe a couple of years old. So if I'm making a kind of particular mod or group here look bad, its situation is probably improved. But you can see the different rules listed here on the left. So some of these are more kind of like more prosaic, kind of like ensuring people are compatible with file format specifications, such as the gene association format. Others are to do with how particular experimental evidence codes are used. So, for example, the inferred from expression pattern code, IEP, should be restricted only to terms in biological process, which kind of makes sense. If you're doing kind of like gene expression, then that's a constraint you want to have here. And we are constantly kind of like adding and deciding and working on new rules. And as we add them, then we can add them to the dashboard and do more automated checks here. So this provides a great way of automating a lot of the consistency checking that we need to do. But of course, not everything is automatable. A lot of this comes down to just a lot of the more nuanced areas of biology and what we do within the Go. So one of the kind of maybe key developments where people don't really realize in the Go over the coming decades is this movement from maybe a kind of motto we used to have, which was kind of annotate to the experiment. And we're moving slightly away from that and being more towards, it's less about annotating the experiment itself and more about annotating the underlying kind of like the conclusions that are derived from those experiments. And doing Go annotation, it's one of the reasons why it's so hard to use NLP methods to automate a lot of this, is it's not simply a matter of kind of like finding the relevant term in the text and associating that with the gene. there's additional nuances here. So, a particular paper may talk about a biological process or something that looks like a biological process. They may mention a term like, apoptotic DNA fragmentation. So that looks like a Go term. But curators bring their underlying knowledge of these genes and the hypothesis being explored in the paper to give more nuanced annotations in the Go. So for example, this first one, if we know that the gene is a DNase and a hypothesis being tested is about the nucleus activity of that gene, then we can maybe make the annotation to DNA fragmentation, apoptotic DNA fragmentation. Whereas in contrast, for something like FOXL2, which is a transcription factor, maybe the correct annotation to make is increasing the rate of apoptosis. And so, as I said before, one of the ways we're encouraging people to think in Go is annotating to the conclusion of the paper, not to the assay. So we're not an experimental repository where people can record what assays were done. The assay is the means to the end, which is trying to understand the in vivo biology of these genes and gene products. So a concrete example here is, you may have an assay that uses a chemical element like rubidium, but what we're not actually interested in rubidium transport here. We're interested in potassium transport, and the rubidium is really just being used as a proxy here. Similarly, with kind of protein kinases being tested with other substrates such as histone, and the goal here is not always to annotate the substrate that was used as in the assay, but to try and understand what the underlying in vivo biology here is. And so same thing with phenotypes. We have a lot of IMP annotations in Go, infer from mutant phenotype, and phenotypes can definitely be used to understand the functions of proteins. But the challenge here is we're actually trying to capture the normal function of these proteins. And when you look at something like a gene knockout experiment or a phenotype experiment, often what you'll see is like not the direct, but the indirect effects of these mutations. So, if you mutate DNA polymerase, it will essentially kind of like, mess up all cellular processes. But that doesn't mean we should annotate that DNA gene to all of those cellular processes, because it's not really actively mediating these processes. It's an indirect effect. The same thing for a lot of kind of whole organism phenotypes, developmental phenotypes as well. If you kind of like knock down some key master regulator or something like that, it will often affect multiple different systems, but it's not necessarily useful to annotate that gene product to all of those different systems if it's not playing a direct role in all of these. So increasingly important are these high throughput evidence codes as well. So, I think, some of the most precise Go annotations come from these more detailed assays where you're dissecting kind of individual gene products one experiment at a time. But, increasingly we're seeing more and more methods that will allow us to, assay the activity of kind of like hundreds or thousands of genes. And so we essentially have high throughput counterparts to the main Go experimental codes as well here. So infer from high throughput direct assay, high throughput expression pattern, and so on. But these are still maybe less used than the main experimental codes. Not everything can always be directly tied to an underlying experiment. So we have these grouping evidence types as well, TAS and NAS, for traceable and non-traceable author statements. And this is where we can, trace back a Go annotation to a statement that's maybe made in the introduction of an article, but we don't necessarily have the full evidence there. And then there's places where a curator just wants to use their own judgment to make an annotation based on, for example, other annotations as well. And we have a kind of catch-all inferred by curator or IC code for this as well. And finally, sometimes you can do a detailed literature survey of all of the experiments on a gene, but really there's no biological data indicating what the function of that gene is, in which case we just assign it essentially to the root term of each hierarchy and give it an ND evidence code. So there's, in addition to these manual experimental evidence types as well, We have a lot of annotations, and particularly in organisms that are maybe not so well, served with actual experiments. We increasingly rely on computational annotations. So there's a number of these. So there's ones to do with kind of like essentially some kind of in silico analysis of the gene sequence, orthology-based methods like inferred from sequence orthology. If you have to kind of like orthologs of genes in different species, you can transfer annotation across from those. There are some caveats though with some of these kinds of methods. There's also things like reviewed from computational analysis, where you've got something like a kind of guilt by association analysis. These also have maybe a few caveats associated with them as well. And then there's also another kind of code that's a bit of a catch-all, inferred from electronic annotation. And this actually encompasses two broad categories of automated annotations. One is kind of, again, sequence-based methods where the annotation is yet to be reviewed. But this is kind of like used less commonly by a lot of the more established model organism databases and human. And more increasingly, the IEA is used for essentially to support kind of like mapping, where annotation has been made into some other vocabulary or some other kind of system and then transferred across to the Go. So I'll give a couple examples of these. This is sphingosine 1 phosphate lyase, a rice gene. And you can see here just a broad spectrum of existing annotations for this gene. You can see here there's a mixture of experimental annotations. That's where you see the manual assertion based on experiment. But we also see a few of these IEA annotations as well. So you can see things like IEA annotations to Swissproc keywords. It's the KW abbreviation. So this is where a first pass of annotation has been done by Swissproc curators using their own vocabularies, and then there's mappings between those vocabularies and Go, and it's been propagated across. But you'll also see here IEAs to Interpro, and not in this particular example, but there's maybe often IEAs that come from something like EC or other enzyme databases. And so a key enzyme database for Go is RHEA. This is a database of reactions developed by the CHEBI group. And what they're aiming to do is to catalog all biological reactions and catalog them in a way where you have very precise descriptions of the underlying chemical elements that participate in those reactions, and they make use of the CHEBI ontology to do this. And so on the RHEA site, you can see how they have kind of like annotated this rice gene in this particular case. So you can see the actual underlying kind of substrates here, sphingosine 1 phosphate, and so on. And so what the RHEA group does is they will annotate a lot of these proteins directly, but they also, the Go group works with RHEA to provide high quality mappings between RHEA and Go, such that we can automatically propagate RHEA annotations onto Go. And we also work with RHEA on mappings to other systems such as Metapsych and EC and so on. And the goal is to have a set of high quality one-to-one mappings such that we can do reliable propagation of annotations across these different systems. And so we've done a lot of work on the mappings with RHEA. And I think this will definitely prove fruitful as well for systems like Metapsych. And I know there's definitely a lot of kind of different kind of like plant species that have high quality annotations using the Metapsych system. So one of the other key systems that we propagate from is Interpro. So we work with the Interpro team on Interpro to Go mappings. And so what this means is that essentially all proteins in UniProt go through the Interpro scan system. They're kind of assigned Interpro IDs describing their domains and families and so on. Interpro incorporates other domain databases like PFAM, and there's been some really kind of exciting work recently on on they have a partnership with Google AI, and so there's kind of new deep learning systems that will give you increased coverage of things like PFAM, and because there's PFAM to interpro Go mappings, we can then do a transitive mapping of these into Go annotations. So this turns out to be particularly useful for those organisms that have fewer experimental annotations. With more well-established organisms, interpro to Go will often kind of like just tell us what we know already, but the annotations here are really high quality, if sometimes conservative. So this is, really useful for, for kind of agricultural species that, don't have such rich experimental data associated with them. And so one of the most reliable ways to propagate annotations across species is to go beyond these some kind of simple pairwise BLAST ISS style mappings, and to actually make use of phylogenetic trees and evolutionary models as part of propagation. And so we provide a number of different evidence types for this kind of phylogenetic annotation. I'll talk about some of these, in the context of the next slide here, but the key one is this inferred from biological aspect of ancestor that allows us to propagate annotations from ancestral nodes in a phylogenetic tree, such as this one. So this is a screenshot of the PAINT phylogenetic annotation system. What this allows us to do is to basically make use of experimental annotations in existing extant species, and then to actually go back and infer the function of the ancestors of these genes and gene products, sometimes billions of years in the past, and then also propagate these annotations back down to existing less well characterized species as well. And these all make use of gene families from the Panther database, and this allows us to actually give us a really detailed picture and make use of actual evolutionary events. So we can see on these phylogenetic trees where not only where genes have evolved, but kind of speciation and duplication events. So you can actually see here in this particular example, you can see the different blocks here representing a duplication event. The top block here represents essentially neofunctionalization. So these paralogs here developed activating enzyme activity, whereas the ones in the bottom block at the bottom all retain the original function of ubiquitin activating enzyme activity. So this is really nice because it can make use of these gain and loss events. So we have a full workflow for this PAINT annotation. You can see on the left, this makes use of the Panther trees themselves. And also crucially, of course, experimental Go annotation from the literature. And it's not a one-off thing. It's a kind of like cyclical thing. The Panther trees are updated in concert with the Quest for Ortholog group every year or so, and new genomes or proteomes are added, all of the time. And then we kind of like regularly run our IBA pipeline where we produce these IBA Go annotations. So you can see the enormous effect this has on some systems such as human annotation. So of course, Go is NHGRI funded, so we're all about making use of model organisms to inform the biology of humans. And you can see here that this what the different circles of the Venn here show contributions of different systems to human annotation, just on experiments that were done on kind of like human cell lines or whatever. That would only give us a kind of small proportion of the annotations we have. But if you also incorporate non-human models, so kind of like everything from mouse to yeast, you can see a much larger circle kind of contributing here. So that's all very well for human, but this is of course relevant to other kind of like species where we may want to make use of model data. So agricultural systems where we want to make use of Arabidopsis, for example. And so there, luckily there's actually a system that essentially extends Panther with a lot more of these plant species. So the Phylogenes project, which is a collaboration between Phoenix and one of the Go PIs, aims to do exactly this. So it extends the existing Panther trees with a whole bunch more plant genomes here, and you can see here how different experimental annotations from Arabidopsis in this auxin family can be used to infer other species such as wine, spinach, and so on. Okay, so one of the main uses of Go is in enrichment analyses. So the basic idea here is that a lot of biology now is high throughput. We do experiments that will, where we'll maybe kind of like, perturb the environment of a plant and look at kind of like changes in gene expression in relationship to biotic, abiotic stress or whatever. And you end up with a big long list of genes, and you want to know what the hell is about this set of genes that makes them different. What's the underlying kind of like mechanism that is at play here? And so it's, unless you have a kind of encyclopedic knowledge of every single gene in your genome and what it does, this is a pretty hard thing to do just by glancing at the gene set itself. So hence this is where Go enrichment comes in. So the idea is that you look for statistically overrepresented sets of Go terms in this gene set. So there's actually a few different kind of like families of analysis you can do here. There's the kind of classic overrepresentation analysis, where you essentially have kind of like a binary set of genes that are either kind of switched on or switched off, as it were. And you can just do kind of like basic kind of things like statistical tests here, like the hypergeometric test and so on. But you may also have quantitative values as well. So if in an RNA-seq experiment, you may actually have some measure of how expressed a particular transcript or gene is. And so there's various methods, from the venerable GCA, gene set enrichment analysis method, to other methods for taking into account those values to give you a more precise set of ontology terms. And then there's methods such as the ontologizer that make use that allow you to make use not only of kind of like frequentist-based statistics, but of kind of Bayesian model-based statistics as well. So whichever method you use, it's really, I can't underline enough the importance of making use of the latest version of Go in your annotation. There's been a number of different papers over the years that show this really makes a huge difference. If Go is a living system, we're constantly improving the annotations all the time. And something that may not have been yielded in an enrichment analysis 5 years ago may be elucidated if you make use of the latest version. And there are a number of tools out there that sometimes don't update their version of Go that they have for kind of 3-4 years or longer. And really you're doing yourself a disservice by using some of these. So always use the tools to make use of the latest version. So there's a number of different tools that allow you to do Go enrichment. Some of them, such as the DOE K-base system, will allow you to incorporate Go enrichment as part of a larger workflow or a larger narrative, such as this Arabidopsis RNA-seq analysis on K-base. But sometimes you don't need to do a complete workflow, and you just want to paste in a bunch of genes into a web-based tool and get back some results. So there's a number of ways of doing this. This is, if you go to the main Go site, you can do an enrichment analysis directly here by simply pasting in your genes and choosing your species. This is all powered by the Panther database system. You can do similar things on other sites as well, and so you can do the same thing essentially on the TER site too, which is more integrated with the TER database as a whole, but it's still using the kind of like the core Go enrichment tool under the hood here. So so you can basically paste in your genes, go straight to the analysis here. By default, it will make use of the complete genome as the reference set, but we always recommend using a background set where possible. This will give you better results. So for example, if you only assayed 80% of the genes in your kind of like, if you're in your expression results, then you should only use those genes that were explicitly assayed as the background set. And of course, in bioinformatics, ID mapping is always a problem. So a good enrichment tool such as this one will tell you how many of the IDs in your input set couldn't be mapped, or how many had ambiguous mappings. In this particular case, there was maybe 40 of them. So that's maybe some cause for concern. So, we recommend always kind of like investigating those. And, sometimes this can involve a bit of manual poking around while we figure out what's going on there. But maybe 40 isn't such a huge area for concern there. And then this will kind of give you back results, with the most statistically significant ones in the top. This is showing a kind of like an inverted tree kind of like view. So you can see the most relevant term here is cellular response to hypoxia, which may be maybe in accordance with the experiment we're doing, where we're kind of like starving the plant of oxygen or whatever. And you can see there's different choices for different kinds of statistical tests, but essentially it will give you something like a p-value, a corrected p-value at the end here. So just moving on to kind of like how Go fits into a larger ecosystem of ontologies. It's not a standalone ontology. It relies on and is relied on by a wide network of other ontologies. I mentioned previously how it makes use of CHEBI for chemical entities to formally describe the different kind of like participants in a reaction. We make use of eco for evidence types, which itself relies on the ontology of biomedical investigations. The sequence ontology is also at the core. And then many ontologies like the human phenotype ontology make use of Go. We make use of the cell ontology, and certainly in mammalian cell biology at the moment, one of the big things at the moment is single cell sequencing and kind of like clustering these and deriving new cell types. And we're making kind of extensive use of Go and kind of helping to interpret some of these experiments. So the underlying representation of Go is something called OWL, the Web Ontology Language. This allows a very precise description of the kind of like the semantics or the axioms underpinning Go. And this allows us to essentially kind of like break down complex concepts like polysaccharide biosynthesis or glucan biosynthesis and turn them into kind of like logical expressions that compose together a computational description in terms of simpler concepts like biosynthesis or polysaccharide and glucan. What this allows us to do is to essentially automatically infer and automatically construct and validate parts of the ontology. So we can make our classification consistent with ontologies like CHEBI, which classifies glucan. Glucan as a kind of polysaccharide. So glucan biosynthesis in Go is also inferred to be a kind of polysaccharide biosynthesis. So there's a lot of other kinds of axioms that we make use of in OWL. We don't necessarily always kind of expose this level of detail to external users of Go, but it's incredibly useful for automatically maintaining the ontology. So I showed a variant of this figure earlier. So one limitation of the classic way of annotating in Go, what we call standard annotations, is that it doesn't really link together these annotations to give a complex picture of how multiple different gene products act as part of an ensemble to carry out a biological process. So, essentially annotations are just a kind of like a big bag with no order to them other than what we may kind of impose in our own minds. But where we actually want to be going is to actually start linking these annotations together so that rather than just being a disorganized heap of annotations, we can actually see the causal flow of activities that go to make up individual biological processes like DNA-directed DNA replication. So it may start with something like the DNA helicase activity and take us all the way through to DNA ligase activity at the end of this part of the process. So we have devised a system within Go for being able to link these annotations together. This is called GoCam, or Gene Ontology Causal Activity Modeling. So we've developed a framework called Noctua that allows for linking together annotations in this way. We can see here three kind of annotons or groups of annotations being linked together. Link RNA binding, chromatin binding, NAD-dependent histone deacetylase, and so on, each in their own individual kind of like cell compartments. So we published a paper on this a couple of years ago, so I invite you to read that if you want to know more about GoCam modeling. Just to kind of show you some of what's going on under the hood is that we essentially have this kind of like more visual biologist-friendly view, where we kind of like group together the Go annotations around individual activities, connect them together causally, but we make use of RDF and the web ontology language under the hood to kind of structure these in a way, in a graph-based representation where everything has its own ID, its own kind of fair IRI, and we can make use of OWL reasoning to make kind of like new inferences over these individual GoCams. And so we're working closely with the Reactome group, primarily with the human pathways in Reactome, but of course there's different instances of Reactome for different species as well, including some plant reactomes as well. And so a lot of this methodology should work there as well. And we've developed kind of essentially transformations to go from the BioPax representation in Reactome to the underlying GoCam models. And this provides us with a kind of like a large bolus of annotations that can go to complement the ones that are being manually created within the Go system itself. So just very briefly, a bit more on how to access the Go, both kind of as maybe a biologist, using kind of like web-based tools, but also as a developer using kind of programmatic methods and so on. So of course, one of the most common ways to access the Go is using dedicated kind of ontology portals, such as the Amigo Ontology Go browser. So you can see the ontology here in the context of different annotations across different species. You can also get kind of complementary views and things like the Alliance of Genome Resources. So this is a relatively new NHGRI funded project that links together a bunch of the NHGRI funded mods to come up with a common framework, and similar ideas to AgBioData here. Because it's NHGRI funded, it's focusing more on kind of like, traditional model organisms here, but, there could be ways to incorporate some of this framework into other systems as well. And in fact, the little ribbon component that you see here at the top is a standalone reusable JavaScript widget, and it has been employed in different databases such as UniProt. So now on the UniProt pages for any individual protein, you can see not only the Go annotations, but their summarization in this Go widget here. You can choose which individual Go Slim to use to sum these annotations up to. And of course, this is a rice protein, so it chooses the Plant Slim by default here. So you can also access the ontology computationally by using either ontology libraries, APIs, or downloading in standard ontology file formats. So maybe I'll just kind of like go very, very quickly over these. So the venerable OBO format was the initial format that we came up with.
Just a time check, Chris. We want to leave a couple of minutes, or maybe about 5 minutes, or at least for questions. Thanks.
All right. So yeah. So just maybe if you're interested in some of the more in the weeds details about some of these formats, come and talk to me. We have OWL. We've got this kind of like newer, more developer friendly JSON format that we're encouraging now as well. We have a Go API that uses the Open API standard for describing how to access both terms and annotations within the Go. There is a new Python library that we're developing that is applicable not just to Go, but across different any ontology within OBO and even ontologies outside OBO as well. It provides both Python access, but it's also got a very curator-friendly command line interface as well that allows you to do powerful operations on ontologies. But for a lot of kind of like classic analysis activities, things like the R language and bioconductor remain the mainstay here. Really just to emphasize that the ontology is not static. We're very interested in kind of working with different communities to improve parts of the Go. In the past, we've had a number of different working groups. We have an ongoing working group that is the multi-organism working group that may be of interest to people here. So we're very interested in things like how genes within individual kind of either pathogenic or symbiotic kind of like microbes, how the pathways within these organisms interact with the pathways of host species such as nicotine plants and so on. And then very quickly, we have a new system that makes it easier for people, we'll be rolling this out soon, a new system that makes it easier for people to request changes in ontology. So you can just now use kind of like controlled natural language to say, make requests for additions to Go terms. You can just do this directly in a GitHub issue, and this will trigger an agent to automatically make a pull request for those changes, which will be reviewed by an ontology editor who doesn't even have to open Protege to make these changes. All right, so maybe I'll just kind of like skip over these. I did want to kind of like give a shout out to the Plantion project that you know, kind of extends on Go's in many ways to kind of like provide vocabularies for describing other aspects of genes and genotypes and so on, with an analogous project for animals, the Monarch initiative that I'm involved with. But maybe just to give us time for questions, I'll just kind of like leave it here, and just to kind of like really highlights the huge amount of work that goes in from different groups, different annotation pipelines to build up the Go. And at its core is a very collaborative project, and we're very interested in hearing from you. So I'll just leave it here with thanking all of the numerous individual databases involved, and also the massive team of curators that go to make Go as successful as it is. And with that, thank you, and I'll take any questions.
Thank you, Chris. Thanks very much for that overview of Go, and it kind of reminds me that the work done by what looks like a massive team, but is still relatively a small number of people, has this big impact on scientists working in kind of all living species. So there are a few questions that I picked, that I copied from the chat. Athy asks about contributing Go annotations. Years ago, Maze GDB started to work with Go on contributing Go annotations. Who is a good contact if Maze GDB or another group would like to do that again?
So I'll follow up with you on that, Ethy. Yeah, in general, though, just the Go help desk is the best place to start. And then from there, we'd have a kind of conversation about what kinds of annotations would be contributed. Are these kind of experimental, or are these kind of computational? We're definitely most interested in kind of like manual experimental annotations.
Yeah, and that's what we would like to contribute. Unfortunately, Mary Schaefer started this years ago, and then she retired before finishing.
I know, yeah, I'd love to just pick up the pieces there again. Yeah, she came to visit us, and we need to pull up there.
Right, thank you.
Great. Then for a couple of questions about the Panther and Paint related work. Lenore asks, what about Panther families that don't have human genes or are plant specific? Do those get curated in Paint by the Go project?
They do indeed, yes, yeah. I mean, there may be within the Go project itself, there may be a bias in which families we focus on, and we, but then if we annotate an ancestral gene that is kind of like, across all, all eukaryotes, then that will get painted down to all kind of like, all extant genes within that family.
Right, but if it's a family that itself does not contain any human genes.
Yeah. I mean, it may be less of a priority for the Paint group to annotate that family, but definitely within the context of the phylogenes project, that could be done, right. And sometimes it will just get done anyway, in which case it will get to the plant genes.
Okay, so following on in the same Panther vein, Andrew Olson asks, when referring to the Panther trees, do ancestral nodes have stable identifiers so that annotations persist when the gene trees update?
Great question. They do. Yeah. They didn't always historically, but now they do. And there are cases where, it's just impossible to transfer one across, and the node may be kind of split or merged or so on. And we've got process for that. But where possible, they do persist across different Panther versions.
So we have a couple of minutes left. I have, and somebody asked about the QC dashboard and the link, and Lenore very kindly posted the link to the dashboard online. Monica, let's take that last question from you.
No problem. Can you hear me?
Yes. And now we can't. Nope.
Don't hear.
Maybe
I don't hear her either.
You can write in the chat, Monica, if you feel.
There's also a question from Andrew Olson.
Right, that was about the identifiers for the ancestral nodes. I think Chris addressed that, unless there was another one.
No, that was the only one I had. Thanks.
Okay, thanks, Andrew. We simply have lost Monica. My question was about, you talked about annotation of phenotypes and how, like in that one case, there was like a glomerulus defect, and that's not really the best thing to annotate the gene product for. Have there been case, or like how has Go been used, or can Go be used for direct annotation of phenotype?
Yeah, so I wouldn't use Go itself. I would use one of the companion phenotype ontologies. So so there's things like the kind of like the trait ontology for plants and the individual kind of CO crop ontologies. They've maybe been used more historically for kind of like, kind of more broader kinds of annotations. But we're doing a lot of work now within the Monarch initiative to essentially unify all of these different ways of doing phenotype annotations and trait annotations. There's a lot of kind of even just deciding on what's a trait and what's a phenotype leads to endless philosophical discussions. But
I think that's where, interaction with AgBioData could also be really interesting and beneficial for the multiple communities that are trying to do similar things with annotating phenotypes.
Yeah, and it will all be linked together such that if you do have a, kind of like, a plant leaf phenotype, you've maybe got genes associated with that, but there may also be some of the same genes may be more directly annotated with, plant leaf development within the Go. So what you end up with is this nice kind of like knowledge graph of all of these different kind of genes interacting together and the different terms interacting together in ontology space.
Yeah. Okay, that takes us over the top of the hour. I know you probably have more questions. Like Chris said, the help desk at Go is one general place to start, but I think, within the community, there are enough connections, and Chris's email is easy to find. You can get in touch directly with him. So I turn it back over to Anarita to close it out.
Well, thank you, Chris, and thank you, Tanya. It was a wonderful webinar. I will thank you all for joining us and see you next month. Bye.
Okay.